hesitate to reach out. We want to be with you the entire way. We want to protect your investment in the platform. We start with this fixation training and the sample prep discussion. I then ask you for some quality metrics as you're preparing your single suspension platform or you're optimizing your protocol. I'll ask you for some metrics, so some fixation quality checks. We'll then schedule another session. We'll go through the entire whole transcriptome process, like how do we barcode, how we library prep. There'll be some QC metrics for your samples while you're doing that as well. And then when you're ready to send for sequencing and your FASTQ files come back, I'll introduce you to our 2 methods that you can use for analyzing your data. We have a cloud-based platform called TrailMaker, which essentially you just upload your FASTQ files. You don't have to know any code, and it'll parse through your data and give you publication-ready figures. We also have a local computational pipeline, which is a local Linux-based platform for those that don't want to use the cloud-based one. That requires a little bit more knowledge of code, but I'll give you all the instructions for that as well. And then we'll have a final debrief just to go through your initial findings, what you see in your data, how your experience was, what your next steps are, and anything that I can help with. So any questions so far?
Nope.
Awesome. So before we get started, just wanted to talk a little bit about your experimental design. So let me actually pull up my notes if you don't mind. I'm gonna stop share. So Brian, I wanted to ask, do you have any current experience with the sample type that you hope to run on Parse? Have you run it on any other single cell platform?
No,
So just have you done any bulk RNA sequencing with these samples?
not with these ones. I did more with like human tissue.
Got it. So you have bulk seq experience, but this will be your first single cell sequencing run.
Right.
Perfect. And so do you have, if you could tell me a little bit about your sample, like we're doing single cell suspension?
Um, yeah, I think so. So basically, we're gonna have mouse brain and lung from either control or environmental exposed or possibly even like high fat diet,
Okay.
and then we're gonna do pump out the perfuse the organs to get rid of the blood, and then harvest the organ tissues. So hopefully, mouse brain will have enough cells for the kit, or the lungs could have enough cells, or single nuclear, however you recommend we do it.
Okay. Sounds good. So when we harvest the organ, like the lung and the brain tissue, do you plan to freeze it, or do you think you'll be able to isolate the cells and fix them immediately
Um, I'm guessing
in terms of
is it better to
workflow?
do the immediate processing?
Yes. Mm-hmm.
Yeah, we could do
So
that.
sorry.
Yeah, we could probably just do immediate.
Perfect. If we do want to snap free the tissue, Brian, we would have to do nuclei. It's going to be trickier to extract like intact cells, from frozen tissue. It's always better to do nuclei, and it helps a little to do nuclei because by the time you put it through that freeze thaw, you've lysed a lot of the cells in that tissue anyway. Yeah, if we could do it so that once your brain and lung is ready, we perfuse the organs, we harvest the tissue, and then just fix it instead of freezing. We just fix it.
Okay.
Okay. Wonderful. And then those fixed samples, like as they come, we can store them at -80 for 6 months. So you have more than enough time to get everything fixed. All the different groups, and then run everything together.
Sounds good.
Awesome. And then Brian, for the single cell protocol, like in terms of like what you're going to do for the mouse, brain, and lung, do you have a isolation protocol already? Do you have one drafted? Or is that something like if you like, I can reach out to other colleagues that have run with fresh mouse, brain, and lung and share some of the considerations with you?
Yeah, that would be better.
Okay. So no specific protocol yet.
No.
Okay. So I'll make sure I'll let you know. And so how many samples, Brian, do you think you'll want to fix and run for your first like pilot
I think
experiment?
we have that WT mini kit, so I think was that 12.
Mm-hmm.
We could either do 6 and 6 or 3 of 4, 4 of 3. So I don't know how what kind of N do you normally have to get a decent amount of significance on these experiments?
That's a great question. So for the mini, it's a total of 12, but it's designed more as a pilot because the total amount of cells we add to the plate is 10,000. So if we do 12 samples, the way it's going to be set up is that 10,000 divided by 12. Each of your samples will only will only barcode about 800 cells.
Oh,
So I think the way we can do it is if we do 6 samples, and that'll give you about 1,600 cells per sample. That's what we usually see. Like people do 6, but we leave that to you. It's more designed as a pilot before you go into our standard kit, which is 100,000 cells.
okay.
Yeah. So like, let's say since we have 3 groups, we have the control group for mouse brain, right? Control group for the lung. So that's 2 samples separate. And then environmental exposure for both the brain and the lung, and then and then a high fat diet. So that's 3 groups total, right?
Up to that. I mean, we would
Uh,
probably for the pilot one would probably just stick with like mouse brain, looking at exposed environmental exposure or not.
yeah, I would recommend. Yeah, let's do brain first. Brain is also the trickier one too, so we can do the pilot on that and see what we um, like how best to move forward with that tissue type. So I would do, let's take the mouse brain tissues. We do the control and the environmental exposure. If you want to pool some brains, do you think you'll pool some brains together, or will you do one individual brain?
I would hope to do just one individual brain. I wouldn't want to pool if we don't have to.
Yeah, I think if we can finalize, if we can optimize that protocol, we should have enough cells for the mini kit to do from one brain. And then you can have 3 samples, and if you want to do it in duplicate, or run just 3, we'll get more data from each of them because it'll be 10,000 divided by the number of samples.
Okay.
Yeah. So let's say, what do you think about for the mini? We designed an experiment with the 3 samples. So mouse brain control versus environmental exposure versus high fat diet.
Let's see. Yeah, the problem with the high fat diet is it takes a really long time
Hmm.
to so those would be like really far distant from each other, I imagine.
Yeah.
But I suppose we could do like a shorter period of time and do like like a pre before they're really sick model and
Yeah.
see if like what the early markers are.
Or Brian, what we could do is, if you want to do the lung as well, let's do control environmental exposure for brain and lung, and that'll give us 4 samples.
Okay, and then that would be like 2,500 cells each.
Yeah,
Okay.
because I feel like that would give you more information for both groups, both the brain and the lung.
Mm-hmm.
If the high fat diet takes a little longer to, in terms of study design.
Sure.
So like if we did 4 samples, it would be 2,500 cells per sample, like in terms of the final barcoding output. So but in order to fix for each of them, so for instance, like for the mouse brain and the mouse lung, so for the each sample, so if we do 4 samples, we'll need a minimum of 100,000 to fix, and I'll explain why. Like I'll go into that right now, when we go through the PowerPoint. But essentially, at the end, we load a little bit extra to ensure that we get that 2,500 cells per sample. Um, in terms of single analysis.
Okay.
um yeah, let me know, like, we have time, so anything that you think of, like, while you're thinking of it a little bit more. I think for a pilot, that would give us a lot of information on how to proceed if we did the brain and lung both. Just do a control and environmental exposure for both of those, run the pilot, like the smaller kit, and that'll give us a lot of information, and then we can decide how to plan a bigger study.
Okay.
What do you think?
I like it.
Awesome. And then, Ryan, how do you plan to count the cells? Like, do you have any? Are you going to use an automated counter? Do you have a manual hemocytometer with trypan blue
I think we have one of the Countess system.
counter system? Okay, if we are going to use the automated, and I know no one likes to count, with the hemocytometer anymore, but I'll recommend, because this is a pilot and because we're going to be taking this fresh tissue and we're going to fix it immediately as soon as we have it harvested, I would recommend we count with the hemocytometer and trypan blue, at least to validate the countess counts. So if you do a spot check, like for instance, one day you're doing both you're doing brain control, right? You're harvesting the control that day. I would recommend that you do a count with the hemocytometer and then a count in the countess, just to double check the count. With the countess, with the Luna, with the Salometer, like those platforms, sometimes they overestimate the number of cells. and then we're not really sure what we're seeing when we actually load the plate. Like we're actually loading less than we think we are.
Okay.
Okay, awesome, thank you. And have you perfused any tissue yet? Like, do you have a rough idea of how many cells you get per brain or per lung?
No, I have no idea.
Okay, so this will be so we're like we can do a little trial run and see what happens.
Right.
Okay, awesome. So I'll put that. And then so we'll fix it immediately. What we can do is then as soon as it's fixed, we know we're not worried about like any batch effects because we can store it and then run them all together. And that way we're not introducing another freeze-thaw where we would cryopreserve the cells, thaw them out, put them through more anything like more, influence them in any other way. But before I continue with the PowerPoint, just final question, Brian, what would define success for you, like with this platform? Like, what do you think is the main goal of the assay for you in terms of this lung and brain tissue? Like, are we looking at differential gene expression between the control and experimental group? Like, what is the main criteria that you think would be most interesting to establish?
I mean, yeah, it would be some kind of unexpected differentially expressed genes in say like a pathway that is not really obvious and would hopefully be publishable with the just with the pilot data, but if not, like usable in the grant.
Mm-hmm. Awesome. Yeah, with the mini, that's also it's usually used, to apply for more grants, but sometimes you do get enough information to like publish something. And that'll be interesting in terms of especially the environmental exposure. When we're exposing them, you said that you're gonna. I know Cooper mentioned briefly, but what is the environmental? Like, are they gonna be in? Like, how are we gonna set up that group?
I mean, like they're going to be exposed over 3 times a week for like 4 weeks,
Mm-hmm.
and then they're basically made to ingest like environmental toxins, and then that kind of travels throughout their system and hopefully gets into their blood and brain barrier and all that.
Sounds good. Week, so we're gonna all that environmental. Okay. So 3 times a week for 4 weeks would be the experimental group, and then the control group, nothing, but we'll keep them also for four
Well,
weeks.
they're sham exposed, so you just give them saline.
Got it. And both of them, like both the brain and the lung groups, they'll be ready to harvest after 4 weeks.
Yeah, something like that.
Okay, awesome. Thank you. All right. So I'll go ahead and share my screen again if you don't mind. Thank you so much, Brian. Sincerely appreciate it. I know Cooper had a lengthy meeting with you, but I always like to double check like what I know and aware of. So I appreciate your time. And so here is just a high level overview of the entire workflow. So what to expect. So we'll take those single cells from the brain, from the lung, and from the control and experimental group. We'll have them in suspension. We'll immediately fix them. So the way that Parse is different from the other platforms is that we're not going to use any equipment. The cells themselves are going to be the equipment, so they're your reaction compartment. So we have to fix and permeabilize the membrane in order to uptake the barcodes. So that's what we'll do with the fixation. After that, each of these fixed cells, in order to ensure diversity and to make sure that each cell gets kind of follows its own path, and like that we can increase the number of combinations of barcodes that each one is labeled with. We will run through 3 rounds of barcoding, and of course we'll meet again when we're ready for barcoding, and I can always come and run barcoding with you guys. It's quite straightforward. It's not tricky. It's just a more in order to make sure that we get as much level of different combinations of barcodes as possible. That fixation process is essential, and also making sure we have a robust suspension. So when you guys do run your first like brain or tissue group, it'll be interesting to see how many cells you get from each one, what they look like. I'll show you some examples of some stains. In addition to trypan blue, I would recommend you at least take a look at what they look like, how their membranes are. We don't want to see any fading or blemming membranes while we fix them, because those will, as we barcode, as we plate to plate, we'll lose the ones that are weak. So when we fix them, we both fix and permeabilize the membrane. So we want to make sure that going in we have strong, robust membranes. After we're done with our libraries, we'll send them for sequencing. When you guys get your FASTQ back, we'll meet again virtually, and I'll show you our different platforms available. I'll actually show you a little bit today at the end, the different resources you have available to you. But essentially, what is the mini kit? So I'll just skip that just to go over the kit in general. So in number of target cells is 10,000. So max number of samples is 12. But like I said, if we do all 12, that'll be about 800 cells per sample. And people, when they're applying for grants, they usually do that, like they'll max out the plate. But sometimes when it's something like this, where we can determine better like how to scale up, they'll do 3 or 4 samples, and that's how they'll scale up. So the first plate will be a reverse transcriptase with the first barcode, and then you'll have 2 additional plates that will be a ligation reactions with the second and third barcode. For this first pilot run, we'll end up with 2 libraries, sub-libraries, and it'll be about greater than 221 different barcode combinations. And we'll go into this part of the process when we're ready for it. So today we're just going to talk about fixation. So the tube-based fixation is a very simple a very simple platform, and it's going to take you about 30 to 60 minutes after you harvest your tissue, so it's not too long. But there are some things to keep in mind before we dive into that a bit deeper, and that's sample and site preparation. So success with the parse assay is going to really depend on our sample prep. So we're going to have to make sure that our cells look really good before we fix them, that they have incorporated all of the things that are necessary to ensure that they survive all the different things that we're going to do to them. So we're going to fix them, we're going to barcode them, we're going to ensure we don't lyse anything until the last barcode. So after the 3rd round of barcoding is when we actually lyse. So there's no lysis that occurs until way at the end. So that's why that fixation process, that sample prep, is so critical. In the guide that I'm going to send you today is a site preparation guide to ensure you have everything before you start fixing. Since we haven't developed a sample prep protocol yet, the first thing what we're going to do is ensure that you guys are optimizing your sample. So if you do want to do like a trial harvesting of a lung or a tissue with some extra, if you have like an extra mouse sample, just to see how many cells you get, that would be good. But I'll show you some things to keep in mind when you're looking at the pre-fixation, like what the sample prep is. But here are some of the different things that'll be in your guide that we recommend that you have, like Mr. Frosty, to make sure that you cool your devices at about 1 degree per minute to minimize cell damage. So as soon as your samples are ready and fixed, we don't want to immediately throw them in the freezer. We want to make sure they cool slowly. We recommend low retention tips, low buy-in tubes. So all of these are laid out in the site preparation guide, and I'll ensure that I send that to you at the end of our discussion today. Additionally, if there's other members of your lab, Brian, that would like to because I know it's the holidays, so if you'd like me to meet with any of them that are going to be dealing with the samples, let me know, and we can have another training discussion with them as well. Like I said, fixation is going to be super simple, super straightforward. There's just some things to think about before we actually fix. So we call these our prefix QC metrics. We want to make sure we have at least 100,000 cells going into fixation. So for each of the mouse brain, the mouse lung samples, when we harvest, we want to make sure we have at least 100,000 cells. 100,000 will ensure that we have enough to follow through to the remaining part of the assay. We also want to make sure we have at least greater than 70% viability, or ideally greater than 90%, but 70 is a good marker. When you're looking at your cells under the microscope, I'm going to ask you to take pictures and examine the physical appearance and structure of your cells. So you want to do a morphological assessment, identify any abnormalities, damage, contamination, ensure that only healthy cells are used for the downstream applications. So we're going to take a look at that when you're preparing your sample prep. Additionally, we're going to add some other metrics. We're going to make sure we have less than 5% of debris. So because we're going to be dealing with fresh cells here, it's important that we some of the protocols that I'll ask my colleagues about, some mouse lungs and mouse brain, and I'll send those to you guys. Removal of debris is critical to minimize clumping. We don't want cells aggregating to each other, so debris is going to ensure that happens. Same thing with aggregation. We don't want doublet rate. We don't want to increase our doublet rate. We want to keep our doublets down. We don't want 2 cells getting barcoded with the same barcode. We want to ensure that each cell adds its own barcode. So we're going to try to aim for less than 5% aggregation. There is a measure that's optional. It's called the RIN score, which is the RNA integrity number. People usually do this more when they're doing single nuclei and they're dealing with frozen tissue. So they'll actually take a slice of the frozen tissue, evaluate the RNA integrity of it. Since you guys are going to be taking your fresh tissue, extracting your cells, and then immediately fixing, I don't think you'll have to worry too much about the RIN score. This is more an optional check that we ask for when we're doing frozen tissue, because the snap freeze aspect of that tissue hinders some of the RNA quality. So you just want to make sure that's as good as possible. And so before we move into sample handling considerations, any questions so far?
No.
Awesome. Thank you. So in your user guide that I'm going to send you, it's going to talk a lot about maximizing sample retention during fixation. So one of those things is we want to make sure we're using protein low bind Eppendorf tubes. So make sure you have low bind tubes, low retention tips. You can also pre-block your tubes with BSA if you want. There's an optional step. But if you're using low bind tubes, you don't have to pre-block. Some other things at the end of our meeting today, the email that I send out is going to have our recommendations for how to incorporate DNase 1 and RNase inhibitors into your single cell prep before you fix it. So I'll send that to you. If you see clumping, we also recommend adding BSA or EDTA. That's something to keep in mind. The final buffer that your single cell suspension is in before you fix it, to reduce aggregation, it needs to be calcium and magnesium free. So that's something to keep in mind. Since we're not worried about adherent cells here, we're not doing any cell culture. We don't have to worry about the enzymes for trypsin, for instance. Trypsin usually has RNases present in it, so we recommend using EEE express instead. DNase 1 and RNase inhibitor is essential in the sample prep because the DNase 1 that removes any sticky DNA, which was when the cells lyse. So we're bound to have some lysis as we're manipulating these cells. So the DNase 1 will help with any of that sticky DNA that binds to each other when the cells lyse and cause aggregation. The RNase inhibitor is essential because in the fixation kit, you also have RNase inhibitor, but that is just enough for the fixation process. For your sample prep, you can use any RNase inhibitor. I'll send you our recommendations for how much to add. We just want to make sure that our molecule of interest does not degrade during the process. So we add that RNase inhibitor. And of course, cell viability is essential. So a lot of the things I'm going to ask you for as you're determining the best sample prep in terms of how you want to harvest these tissues is you want to look for greater than 70% viability. You want to look at the morphology of your cells. You can use a couple of different dyes. Of course, trypan blue, calcium AM, which stains live cells. We have AOPI, we know which stains live and dead cells. If you see in your fresh obtained cells from the organ that you see a lot of dead cells, we have some commercial dead cell removal kits we recommend to get rid of those dead cells and increase your viability. But essentially what you're looking for is a single cell suspension that has minimal debris and aggregation. So this is just a small example of something that looks good. While some of these cells are closer to each other, they're not clumping yet. Whereas here you can see that they're clumping together. So this is something definitely where I would ask you to resuspend a little bit better before you take the count. Here you see some debris. If you see more, lots of debris, sometimes this is just trypan blue debris. So it's always good to spin down your trypan blue before you make your count. But these are just some examples of what to look for when you're working with various types of sample prep. You want to not see these chunks of clumpy cells. You don't want to see any debris. You want your cells to be as separated as possible, and you want that suspension to be as clean as possible. And again, if you want to use some other dyes, you can use AOPI. It stains dead cells as red, live cells as green. Calcium AM only stains live cells. Trypan blue only stains dead cells. After you fix, you're going to take a post-fixation count and everything is going to be blue. So don't worry. Sometimes I get a panicked email like, hey, I just did my post-fix count, everything is dead. Well, yeah, because we fixed it, so it's going to be dead. So don't worry when everything is dead at the end. But in the beginning, it's good to take a look at your sample. Just establish its pre-fixation count and viability, and then post-fixation its count, because viability, of course, is going to be, everything is going to be dead. Here are some images about evaluating nuclei. Since we're not working with nuclei, we're working with cells, I'm just going to skip through. The idea is the same. It's just with nuclei, it's harder to establish a number viability, so morphological staining is more essential with nuclei. So we recommend things like DAPI, cyber green, some fluorescent stains to help establish that. Make sure you don't see any chromatin condensation, anything that's blebbing. You want to see round, smooth borders. So these are just some slide examples for nuclei and from tissue. This is an example of frozen mouse brain tissue. So since we're working with mouse brain here, this is frozen tissue and then the debris associated with that frozen brain. So when the brain was snap freeze, and then I took it out, and there's a lot of debris in that sample. So I did OptiPrep, which is a density centrifugation. There's sucrose gradient centrifugation. There's a lot of different types of centrifugation that helps with cleanup. So this is just an example of that same nuclei, but after the density centrifugation cleanup. So again, this is nuclei, but just wanted to show you that there are some options for cleanup if you do see a lot of debris from some of these tissue types. Again, a pre-fixation quality check that's optional is that RIN score. If you wanted to take 100,000 cells from your live single cell suspension and run this, feel free. You just isolate total RNA and run a trace just to see how good the RNA is. So we look for a RIN score of greater than 8. And this is more of an optional check, but for frozen tissues, I always recommend this before you get started because some of those tissues may have been stored for a really long time. If you do decide, Brian, that it's easier workflow-wise to freeze the tissue, then we would have to do nuclei fixation, and then it would be good to get our RIN score just to see if that freeze thaw affects the tissue in any way. And we covered this earlier, and I thank you so much for being open to validating the automated count with your hemocytometer. I know no one wants to count manually anymore, and I completely agree. It's easier to just load our countess slide. But what we found is when you're working with bulk sequencing and bulk samples, it's okay if the count is under or overestimated. But with single cell, single nuclei, each count is very essential. We need to incorporate validation during the fixation, both pre and post, because we want to make sure that that automated counter is counting cells and not debris, not aggregation, and over inflating our count of interest. So that's how we always ask that you make sure that you validate your automated count with a manual count, just to make sure that the automated counter is counting as close as possible to the manual count and not including debris or aggregation in your count. There's 4 major counts in the parse protocol for each sample, but for right now, since we're talking about fixation, there's 2 counts. It's going to be your pre-fix live count, so the count we obtain at the end of our harvesting when we have our single cell suspension, that count, and then our post-fix count when we're ready to store it after fixation. So these 2 counts are really essential because they'll help us establish a retention rate. We're bound to lose cells as we fix them, definitely, but this will help us establish a retention rate, which will help us assess how best to load this first plate, so that when we thaw out this fixed sample, we're going to do another count, and we're going to update a sample loading table. I'll show you guys what I mean by that, and of course we're going to go into that in more detail when we're ready for section 1, 2, and 3 of the barcoding process, so that's later on. In terms of just fixation, though, that you're going to do first after sample prep, just make sure you take pictures of each sample pre-fixation, get your count, take photos post-fixation, get your count, and that will help. And that's all for sample prep. Any questions?
No, it seems pretty straightforward.
Pretty straightforward, yeah. And then Brian, I'll send you some of the different protocols that I'm aware of that people have used for mouse and brain and lung. And just so that if you'd like to take them out, if you'd like to check them out, if you do decide to do another protocol or to work on a homebrew, let me know, and I can help you with that as well. Anything that I can help you as you start preparing your single cell protocol. And okay, so for fixation, now we're actually going to talk about the parse part of so we haven't opened anything in the kit yet. So once we're ready to open the kit, this is the protocol for fixation. Like I said, it's going to be very straightforward. The thing with fixation, like one of the major popularities of this protocol, is the fact that we can fix and then run them later. So same thing here. Like if you wanted to work on the brain first, get those samples ready, then do the lung, and get those samples ready. It doesn't matter if we fix and then freeze them, and versus if we had fresh fixed and run them immediately. There's no major difference between those that have been stored for 6 months at -80 versus those that have been fresh fixed in terms of the genes and transcripts per cell. We've done this we've established this on a bunch of different sample types. But I believe this example right now here is from PBMCs. So this whole Evercode platform is less susceptible to batch effects because of this fixation process. So because samples can be fixed at a certain time, that biology is kind of locked in. Once the samples are ready, they can all be analyzed together. Thus, we minimize batch effects. We make sure that like if you're running the sequencer isn't ready for you until like 2 weeks from now, that's okay. It shouldn't affect our gene or transcript level because by the time we barcode it, we're going to be using our fixed samples. So what does the workflow look like for fixation? So this is an original slide that I showed you. It's composed of 3 major steps. The first one is you take your 100,000 to 1 million single cells, so our cell suspension. We centrifuge, and now this has a range in the protocol, and in every step of both fixation and later on when we talk barcoding library prep, we give you a range. So we give you 5 to 10 minutes at 200 to 500. However, the range is there because we want you to establish and optimize the centrifugation speed that works best for your sample type and use that same speed and time for fixation, for barcoding, for library prep. So we're going to carry that through. We're not going to change that in any way. So we're going to use that same one that keeps those samples happy. So you're going to do that here. Like let's say for these lung, we harvested the lung tissue, we got our single cells, and we think like we were spinning at 400 G. So that's exactly what I'm going to do here. I'll spin at 400 G, remove the supernatant, and then resuspend in our master mix. This is to buffer swap, so we're going to exchange the buffer here, do a cleanup, and we're going to strain into a new tube. So this is the first part is cleaning it. We're then going to fix, permeabilize, and neutralize. So we're going to make sure we fix our cell membranes, then permeabilize them, and then stop that reaction before we once again clean the sample. And then we store them, and we're ready to thaw them out and run them with barcoding library prep once we're ready with all of the samples. The kit itself, the fixation kit, comes ready with 12 reactions, and those bulk reagents can be freeze-thawed up to 4 times. However, to maximize your use, if you wanted to make aliquots of those bulk reagents, there's instructions in the user guide to do that, so you can maximize the amount of times that you would freeze-thaw them. But the master mix, each day that you're going to be fixing, you would make that master mix fresh and use it that same day. So that's why in order to maximize the freeze thaw, if you wanted to create aliquots of the bulk reagents, please feel free. But if you're using those reagents in the box, they can be freeze thawed up to 4 times. All right, so let's go through this step by step. The first step, we're going to centrifuge, remove our supernatant, strain into a new tube. And I apologize, a lot of this is going to seem very unnecessary, but we have a lot of people that are more molecular biology experts. They don't work a lot with cells. So these videos were made more for them, and you guys are more familiar with cells. But essentially, when you're going to remove your supernatant, we're not going to use a vacuum aspirator. We recommend using a pipette, so your 1 ml pipette. You're going to carefully remove your supernatant, and then here she's putting it in the waste container. However, I would recommend save your supernatant. If you take a count and you notice that you have low count or you're losing cells, it's always helpful to look at the supernatant and see, am I losing it to just spinning? So something to keep in mind. So here we're putting in waste, but I would recommend actually keep it. In the protocol for both this part and then later when downstream, it's going to tell you always to remove supernatant, but leave 20 to 40 microliters of liquid on the top. This is just so that you don't lose your pellet. This step here is going to facilitate the buffer swap, but also eliminate any ambient RNA and small debris from the sample before we get ready to fix it. And then next, after that, we strain the cells. So following resuspension, we want to make sure that we remove any aggregates. So we spin, resuspend in the cell prefix master mix, and now we're going to strain it. So when we do this, there is another video, and I apologize if this seems pedantic. We want to make sure that we maintain contact on the mesh of the strainer and literally like plunge everything through. So depress your plunger quickly and steadily without allowing any volume to pool, because you don't want to lose any cells in around the strainer. So you don't want to let them pool, when you're working with bulk samples, it's not that bad, but here we're working with single cells, we're working with smaller volumes, so we want to push everything right through. And in order to do that, we have tested a wide variety of strainers. We have 4 recommendations for strainer types that you can use. So the purpose of the strainer is, of course, to grab any aggregates, multiplets, doublets in the mesh, and then just allow single cells to flow through. So we do have some recommendations here. Different types in the site preparation guide I'm going to send you for fixation. There's going to be some these are going to be listed out there in detail, like with catalog numbers. But I would recommend using one of these. There is a very popular strainer called the Flomy strainer, which is a really great strainer, but we found that it gets clogged when we use our fixation concentration, so I would recommend we don't use the Flomy strainer. So there are some recommended strainers and filters, so I'll send that site prep guide to you and it'll show you. After you filter it, you're going to add your fixative, incubate on ice for 10 minutes. Then you're going to add permeabilization solution, incubate for 3 minutes on ice before you add your fix and perm stop buffer, centrifuge again. When you're adding the fixative, again, this is another pedantic video, but we only want to mix 3 times. Research and development and the product development team found that when people were mixing more than 3 times, we were irreversibly fixing cells to each other rather than fixing the suspension as a whole. So here you'll see she's going to pull up her fixative and then add it to her single cell suspension. So we add it in, and then we're only going to go up and down 1, 2, 3. So just 3 times. Again, this is just to ensure that the more we mix, what happens is we're now fixing the cells irreversibly to each other rather than fixing the suspension as a whole. All right, after we add the fixative, we add our permeabilization, we stop that reaction, we centrifuge again, filter again, and we're going to store the samples after we count them. Now, the cell storage master mix, when you resuspend it according to the protocol, it's going to ask you to count your fixed cells. The reason being the post-fix count is very essential because you have a minimum concentration needed to run the assay. So for the WT minis, the pilot study, post-fixation, we need at least greater than or equal to 500 cells per microliter. Why? Because when we thaw that out, even if we lost like 40 to 50%, we would still have enough to fully load the first plate of that kit. So for this mini, for each sample, you need about 298 cells per microliter to load it. But that's why this target post-fixation concentration is so essential. When you count that last count, it helps us track and ensure that we have enough cells to go through the rest of the downstream applications. And then later, when you're aliquoting in the guide, it's going to ask you to carefully assess your final volume and then make your aliquots. I would recommend, and I'm going to, of course, send you these slides, that you create a counting aliquot and then 2 bulk aliquots, simply because for barcoding, since we're running a pilot study, we're not going to be counting a lot the day of barcoding. But if you were running 12 samples, 16 samples, 20 samples, etc., it helps to have these counting aliquots, which you can count a day before, 2 days before, just thaw them out, count them, upload the sample loading table, which I'm going to show you now to establish the experiment. And then that day when you thaw out the bulk sample, it's not sitting on ice while you're counting the others. So like it just immediately goes into processing. So that's why in the guide and here in these slides, we always recommend you create a counting aliquot of the fixed sample, so at least 10 microliters to count. And so pre-fixation and going into fixation, there's some things to keep track of. When you're running with parse, in order to protect your investment, I will reach out to you to ask you for certain things like your number, like how is it going, you know, how is the sample prep going, is there anything I can help with. But when you do start harvesting your tissue and getting your counts, please share images with me, counts with me of the cells before fixation, after fixation, and those images, it helps us to establish the progress of the assay to ensure that, you know, we can assist if you see anything weird or if you see things that aren't working well. We want to make sure that we support you. And so those are some things to keep track of. In the email I send you as a follow-up, I'll also list those out. Once we're all fixed and ready to go, then we will meet again and we'll talk about barcoding and library prep. But very quickly, before I take up more of your time, I'd like to show you that sample loading table. We will talk about that in more detail once we're ready to barcode, but just as an introduction, on our support suite, so I'm going to ensure that you guys have access to our support suite. You're going to have a customer login, so you're going to create an account, sign in, And you're going to have access to a bunch of different things, including our data analysis platforms. But essentially, here's the user guides, and you'll see your self-fixation user guide here. When we're ready to run the mini, those user guides are here. But if you scroll down, there's the EverCode sample loading tables. So we're going to be running the mini kit. So there's a video that guides you, but I just like to show you today. So if I open up this sample loading table, enable macros, and I'll actually save this and send this to you via email just so that you have a copy as well. But I just wanted to make sure I showed you where to find it. Um, so this is the first plate of barcoding. This is the table that's going to establish the concentrations to load that first plate. So it does all the math for us. So let's say we decide we decide on 4 samples total. So step 1, ensure macros are enabled. Step 2, input the number of samples. So that's number 4 samples. You'll see that I'll get this warning here because right now I'm establishing that the first sample is 100% of my library. That's not true anymore. Each sample is going to be 25% of my library. And so the target number of barcoded cells per sample is going to be 2,500. What is the minimum concentration needed to load the plate? 298 cells per microliter with an overage of 10%. So let's say we thawed out the first sample, the mouse brain sample, and we got 560 cells per microliter. So then this would tell us exactly how to dilute that sample. So the volume of the sample stock solution and the dilution buffer. We would create this and then load the plate. It's 14 microliters in each well, and the second tab shows you that. So the 12 wells, this is how you would load. It's 3 wells per sample, but each of these wells has a different barcode. So every time you're adding to a plate in parse, you will be changing your tip, even if it's the same sample. So separate tip here, separate tip here, separate tip here, because each well will have a different barcode. And so we'll do that for these samples, but this volume here is enough to add 14 microliters to each well. So that's why this sheet was set up, as well as that overage, because we want to make sure that we load enough cells to run through all 3 plates. And yep, that's the sample loading table, and we'll of course cover this in more detail when you guys are ready to start barcoding. But right now, since we're working on sample prep and fixation, I think it would be best to harvest the fresh tissue, get your single cells, don't cryo freeze them, just fix them. And then that just increases viability down the line. We don't have to worry about any freeze cycles. And then once we're ready for barcoding, we'll go over this table again. But just so that I explain a little bit about where those concentrations come from. Any questions?
Nope.
I hope this was helpful. I apologize that I took so much of your time the day after a holiday.
No, it's fine.
Yeah.
I can't like walk anyway, so it's fine.
Oh no, what happened?
I just have I think I have to have surgery on my ankle.
Brian, I have, is it tendonitis?
Bone spurs.
Bone spurs. Okay, my right ankle tendonitis. I feel your pain. I know it's not as bad as bone spurs, but it sucks.
Yeah, I had to
Yeah.
get an MRI today, so hopefully they'll be able to
Yeah.
see how much needs to be taken out, and then I can get it. I don't know, like hopefully it won't be too invasive.